Can you hear me back there? Yeah. Okay. So I'm going to present to you a phylogenetic analysis of the covariance structure in the skull of Anthropoid primates. So, morphological systems have a tendency to covariate due to genetic control developmental interactions. In the mammalian skull, for instance, development is a series of developmental processes are overlapping in producing adult phenotypes. And the variation in timing, rate, scope, and rhythm of these processes structure covariance patterns. And of course, all these developmental processes are under genetic control. Makes sense? Okay. So, covariance structure is also affected by the processes of canalization, developmental canalization, and stabilizing selection over developmental systems themselves, or through the functional interactions in the resulting adult phenotype. So if you think development as this rolling ball over here, this rolling ball cannot go through other paths than those depicted by the ridges, the values of these mountains. Okay, so we expect that changes in covariance structure between sister lineages will display a non-random pattern consistent with these developmental and functional interactions, right? So, let's try it out. Investigate. So here we investigate the evolution of skull size and shape covariance structure in Anthropoid primates in a phylogenetic framework. First of all, let's meet our monkey friends. We have a sample of 5,108 individuals distributed across 109 species of Anthropoid primates, and the species depicted here are distributed across all major clades above the genus level. We don't have all genera represented here, but most tribes and subfamilies are represented. And what kind of measurements we did? We represent our size and shape of our structure using log centroid size for size, right? And we represent shape using local shape variables, which were well, what local shape variables are? They are log volume transformations calculated from, for each individual in the sample, calculated from a reference shape using TPS function. So, and we calculate this local shape variables in the midline of each one of these distances, right? So each individual is represented as 6 landmarks, and we estimate 38 local shape variables in the midline of each one of these distances here represented over a marmoset skull, lateral and ventral. Right? So these traits are grouped according to both developmental interactions and functional interactions in these 6 regions: oral, nasal, orbit, vault, zygomatic, and base. Right? And these regions here are used as a framework for us to understand changes in covariance structure related to developmental and functional interaction. So, how do we estimate covariance matrices for each one of these species? We use linear models to correct for fixed effects of little interest in this context, so such as sexual dimorphism and subspecific variation. And to estimate covariance matrices using these models, we are working under a Bayesian framework, which allows us to estimate a posterior distribution of P matrices that allows us to represent uncertainty in estimating these matrices. And it also allows us to estimate uncertainty regarding any other parameters that are calculated over these matrices, right? Okay, this is a depiction of the phylogeny I work here that has been published in 2012 by Springer and collaborators. And these are the models, linear models corrected for each species. Okay, so moving on. First of all, how to estimate changes in covariance structure. We used a covariance matrix, diversity decomposition over the phylogeny. So here is the space where matrices lie. Covariance matrices are really weird mathematical objects, right? So they exist in a non-Euclidean space. We estimate distances, non-Euclidean distances among these matrices, and we use these distances to decompose these distances on the phylogeny, looking for the distribution of matrix diversity over the phylogeny. And we use a series of randomization tests to test things about the distribution of these diversity over the phylogeny, such as if the diversity is concentrated on a single node, or if it's concentrated on a few nodes, or if it's skewed towards the root of the tree or the tips of the tree. Furthermore, in order to describe such matrix variation, we use the eigentensor decomposition that has been proposed by Hein and collaborators in 2006. And what we do is first we take the matrices that are lying here in their weird non-Euclidean space, and we project, we map them to an actual Euclidean space. And by using a principal component analysis of the covariance matrix among covariance matrices, so it's kind of PCA on the covariance matrix, right? We can produce an basis for this space, in the form of principal matrices. So this is like an axis of matrix variation, right? And we use the projections of these matrices over these axes to do another principal component analysis. But this time we account for the phylogenetic structure associated with this taxon. So it's kind of a reorientation of the space that structures the space, accounts for the phylogenetic structure of the space. So we have a set of global PCs that describe changes more skewed toward the root of the tree, and we have a set of local principal components which describe variation closer to the tip of the trees, right? And we can differentiate between these two types of principal components, as I'll show you later. Okay, so in order to describe what do these axes represent, we take the upper and lower limits of each one of these axes, as represented by their empirical 95% confidence intervals, and we reconstruct the matrices and put them back into their non-Euclidean space. And in order to actually describe variation associated with each one of these axes, we did a selection response decomposition, which allows us to pinpoint which traits are changing associated with each one of these axes. And since we have a posterior distribution of differences in trait specific, we have a posterior distribution of matrices, we can estimate a posterior distribution of differences in trait specific covariance structure. Okay? So, first of all, matrix diversity. And as you can see here, most of matrix diversity for anthropoid primates is in the initial split between new world monkeys and old world monkeys, right? But that's kind of like 10% of all diversity. The rest of the remaining 90% of diversity is skewed towards, is distributed along the phylogeny. And okay, so this is the distribution of eigenvalues for the phylogenetic PCA, and we have a set of global PCs and a set of local PCs. And I'm going to show you quickly now the first two global PCs and the first two local PCs and what kind of variation in terms of covariance structure they represent. So the first global PC represents the split between newer monkeys and old world monkeys with hominids in the middle. And the pattern that they describe is stability in both oral traits, nasal traits, vault traits, and lots of differences in both orbit and part of the yellow one here, and basic cranial traits, right? And remaining PCs, the second global PC represents contrast within new world monkeys and old world monkeys, and the pattern of changes in traits are the same. So oral nasal traits are stable in terms of covariance structure, and orbit and basal traits, basic cranial traits are changing in terms of covariance structure. So local PCs represent contrast between sister species, and well, the first one and the second one, and they also represent changes in terms of orbit and base traits. Okay. So, first of all, matrix disparity is weakly associated with divergence time. So we have more changes in the base of the tree and less divergence along the tips. And some regions maintain overall the same covariance structure, such as the arm region, nasal region, vault region. And other regions display a consistent pattern of changes. Basic cranial, orbit, ocean. I didn't tell you about the same inside. But they also change in terms of covariance structure. These regions, you may think of them as tightly integrated in terms of both functional and development interactions. But the other regions may have you may understand them not as tightly integrated through development and function, but with some sort of heterogeneity, especially in the basic cranium, since the basic cranium is assembled through tissues of multiple developmental organs, and it displays a complex. It is the first region to ossify, and it also has a complex pattern of growth and resorption during development. Okay, so that's it. Oh, yeah. Thus, the pattern of changes in covariance structure and anthropoid diversification is definitely not random with respect to functional and developmental